let's go ahead and get started. There's only one left in this chapter, but I think that's all that I got. It's all the farther that I got. So I'm looking at this slide again because it is related to exit activation in mammals, so our mechanism of dosage compensation. So I said last time that females have one X chromosome active in each of your cells, but that inactivation is random, and it happens sometime after you're a one cell embryo. So some of your cells will have one X inactivated, and some of the cells will have the other X inactivated. And so the beautiful tortoiseshell or calico cats. Surprisingly, as a geneticist, and having had any number of cats, I've never had a calico or a tortoiseshell cat. I think I need to get one at some point. But we're loaded up with animals right now, so have to wait. So coat color in cats, at least what we're talking about here, is the result of two alleles on the X chromosome. One of them is for black fur, one of them is for orange fur. I would assume, as it normally would be, that black fur is dominant over orange fur, but it really doesn't matter in this case because these alleles are on the X chromosome. So I used to put XB and XO for black and orange, but the book has plus for black and O for orange, and I don't want to confuse you guys. So you can do either way. So X plus is black, XO is orange, but X plus XO ends up being a tortoiseshell because females are mosaics for the X chromosome. You can see there's patches of black fur and patches of orange fur. In some cases, like this one on the right here, she's got a lot bigger patches there. Now this striping is other genes, so don't, we're just like black or something else like the orange. Whereas this one is more of a thinned out a little bit. So it just results on when the X gets inactivated, and then after the X gets inactivated, every mitosis after that, it doesn't change. It's not like the cell randomly flips a coin each generation. So you get what we refer to as clones of cells. Like this whole patch here started as one cell. This whole patch here probably started as one cell. There could be some exceptions to that. There could be two cells here, another cell up there. Yes? That's right. So, do you have one? Have you had one? So, that's why I said she, just looking at this, right? I didn't lift up the tail and have a peek, but except for rare instances, all tortoiseshell and calico cats are going to be female, except instances where you got it if you have an XXY. So anybody you know who has a male tortoiseshell cat or a male calico cat, they have a Klinefelter syndrome cat. My postdoctoral fellowship mentor had one of those and just gushed on and on and on ad nauseum about how he had a Klinefelter cat as a kid and didn't know about it. But anyway, there you go with that. All right, so as always, can't have something we talk about without having a problem to reinforce it and help you understand. So, female tortoiseshell cat is, so males, because they are XY, they're, except for, so we're going to say no to the XXY for what we're talking about here. So males only have one X, so they're going to be a solid color. Either they can be black or they can be orange. Now we're talking about the black and orange alleles. I know there's gray and there's the tabby where you get the tiger stripes. That's another gene that breaks up the coat color and makes the tiger stripes, and there's all kinds of stuff, but we're just talking about these alleles here. All right. So female tortoiseshell cat is crossed with a male black cat. What will the coat coloration of the kittens be? So the female is going to be XX, and she is tortoiseshell, so X plus and XO. And in this case, we said the male is a black cat, so he has to be X plus and Y. So don't make the mistake of giving a black or orange allele on the Y chromosome, because this is an X-linked phenomenon, not a Y-linked phenomenon. So then you just fill out the Punnett square as normal. And here's what you get. So for the males, their coat color is always going to be solid, and it's going to be determined by what color they get from the female cat. And so in this case, because the female is tortoiseshell, she has two different alleles to give, and one half of the male progeny are going to be black. That's the top one here. And the other half are going to be orange right there. Then for the females, one half of the female progeny are going to be solid color, matching the color of the male, and the other half of the female progeny are going to be the tortoiseshell. Make sense? Now, if it was reversed and you had an orange cat, then the males would still be half black and half orange, but in the female cats over here, instead of this one being solid color, this would be the calico, because it would be X plus XO, and this daughter down here would be XOXO. So you always get half solid color females matching the color of the male, and half tortoiseshell or calico cats. Not going to get into it because we don't have, there's too many genes or whatever, but this cat is a calico cat. They also have piebalding. Piebalding is a separate gene that makes the face and mostly the underside. If you flip this cat over and didn't die in the process, it would be almost entirely white underneath. And so the piebalding is the same allele, same piebalding allele that's involved with the coloration of beagles. So like beagles for dogs are like that same thing, where they're like all white underneath. And this is the tortoiseshell up here, but you can see this one must have had the X inactivation event take place much earlier, because she's got a lot of big, patches of black and white, or of black and orange. It's not really dilute. And you can see kind of in here too that, like in the orange, she's got the tabby allele, so that makes the tiger striping, but when you are solid black color, you can't see that. Actually, we've had a few black cats where the fur color isn't as dark as it could have been, and one of them we've had where you could actually see, if you put him in the right light, that you could actually see striping on him. This cat liked to sit in the sun, and so his fur would get a little bleached out in the summer, and so you could see the tabby underneath there. All right, so here's your thought-provoking question that will require you to remember stuff from before. Here's the tortoiseshell, right? We just talked about. Remember back when we did the fruit flies? So this male has white eyes because he's got the white allele, and males are hemizygous for the X. But here you have an X plus XW. Yet that eye is 100% wild type, like the red color. There's no patches of white in there that are too small for you to see them, or individual cells, or anything like that. Every ommatidium, or little eye facet in here, is the red color. So thinking back to how fruit flies and mammals differ in how they do dosage compensation, why would this fruit fly eye in the female not be mosaic just like the cat? They do not have X chromosome deactivation. Who can remember, because the test was just Thursday, what the mechanism, and I asked a question on it, the mechanism for dosage compensation in fruit flies? Hypertranscription in the male. So the two X's in the female both stay on at 100%, but the X in the male transcription is, or the transcription of the genes is jacked up to 2 times the normal level, so you get 2 doses here, 2 doses here, and you're fine. So this is where a difference in the mechanisms of dosage compensation can lead to differences in phenotypes there. All right. Now, we are exiting chapter 4. The end of everything made sense. And we're going to enter chapter 5, where I tell you all the things that I lied about previously. Actually, I didn't lie about anything. In chapter 5, we're going to talk about extensions and modifications of the basic principles. So things that happen with a phenotype that you wouldn't expect based on what we've talked about so far. But the very, very important thing to remember is that the inheritance of the chromosomes does not change. The differences we're going to, a little bug, the differences we're going to talk about here are due to interactions of the alleles. So the inheritance of the alleles is exactly the same. You're going to see how Mendel was lucky in that he picked what was going on. So here, we'll get to this later on, but you can see that these two snails have a difference in coiling of the shell. So this is behind and this is coming out toward the front. So you can see that this shell is coiling in a left-hand format here, and the shell of this snail is from the back to the front is coiling in a right-hand coil, and we're going to see coming up to later exactly why that's the case. All right, so previously we've talked about dominance, and we've said, I've made sure to say complete dominance, so that there's one allele that is dominant over the other one. If you look here, you have a red flower and a white flower. So if red is allele number 1 and white is allele number 2, obviously if you're homozygous for one of them, you'll be red, and if you're homozygous for the other one, you'll be white. One of them is going to be dominant. If red is the dominant allele, then having one red allele is enough to make you completely red, as red as you would be in a homozygote. If white is completely dominant, then a heterozygote having just one A2 allele would be as white as the one up here. There would be no red in there at all. So one allele in this case with the red dominant, and one allele is enough to show full pigment, and here there is not enough pigment produced in this case to show any color in the flower at all. All right, now we're going to venture into incomplete dominance. So incomplete dominance is when you actually get a mixing. So you get not enough pigment to be fully red, but enough to differentiate from white. So here's fully red. Well, this one isn't fully red, and this one, you can see there's a range. Back here, see, this is the dark red. So A1, A2 would actually be some kind of intermediate, because you're getting some red pigment produced, but you can tell that there's not enough red pigment to mistake it for the homozygote. Let's use the eggplant example. Who wants an eggplant parmesan, right? So if you look, a lot of the eggplants in the grocery store you'll see, and they're sold, are like these very pretty dark purple ones. they've also got some white ones. And purple is dominant over white, and you get a big P allele. White is little p allele. And then we're going to cross these together. They're both homozygous or true breeding. So the F1 generation is going to be big P, little p, right? If there was complete dominance, what color would all the F1 progeny be? Yeah, purple. The dark purple is, they would be as purple as this one. But in this case, you have incomplete dominance. So now what are you going to expect? Right, some kind of a lavender or light purple color. You got it. And that's exactly what, I mean, they're calling it violet, but that's exactly what it is. So with one big P and one little p allele, you get enough purple pigment produced that you can tell it's not a white eggplant, but you get only half the dose of whatever the gene for purple color is doing here, so that you end up with something in the middle. With incomplete dominance in this way, you're actually going to see when you do the cross that now, because you can tell the phenotype of the heterozygote from each of the homozygotes up here, that your 1 to 2 to 1 genotype ratio is going to be the same as the phenotype ratio. So when we cross these guys, they both give the same kind of, the alleles. You can have a big P or a little p, and you're going to get your 1 to 2 to 1 genotype ratio, but you're also going to get a 1 to 2 to 1 phenotype ratio. So when you do a monohybrid cross like this with only one gene involved, you can tell whether there's complete dominance or incomplete dominance by whether you get in the or when you cross two heterozygotes in the progeny of that cross, if you get a 3 to 1 phenotype ratio, then you got complete dominance. If you get a 1 to 2 to 1, then you've got incomplete dominance. Note that in both cases, complete dominance or incomplete dominance, it's so many out of 4. So here it's 1 to 2 to 1. That adds up to 4. Or 3 to 1. That adds up to 4. We're going to find out coming up that there are cases where one of the squares on the Punnett square are going to drop away. Like when we talked about the disease alleles and said, could this person be a carrier? And we had to eliminate the square on the Punnett square that was homozygous for the recessive disease allele, because that person would have the disease and we wouldn't be counting them. All right, so complete dominance, incomplete dominance, and now we're going to talk about codominance. So codominance is when there are two alleles that you can tell them apart, and you can tell if one of them is there, the other one is there, or if both the first and the second are there. And the example that you've undoubtedly seen before, but maybe not considered it in this way, is the human ABO blood type antigens. So these are little sugars that are present on the cell surface of all of your red blood cells. Well, unless you're type O like me. So the A antigen is coded for by, this is a big I, not an L. So big IA. The B antigen is coded for by big IB, and little I is recessive. So big IA, big IB are both dominant, and little I is recessive. Little I is recessive because you don't have any antigen on there at all. If you are blood type A, because big IA is dominant to little I, you can be either heterozygote big IA or homozygote big IA or heterozygote big IA little I. Same thing for type B, homozygous big IB or heterozygous big IB little I. Here's the difference. If you have one big IA allele and one big IB allele, you have codominance because there's room on the cell surface to express both antigens. It's not completely covered. So you can tell if both antigens are present. The presence of the A antigen, and I'll tell you, if you look with a strong enough microscope, they really do look like little blue triangles and little green hexagons. Is present, and the B antigen is present. One of them does not interfere with the other one. Then if you're blood type O, you have to be little I because the A and B are dominant, and you would just have nothing on you would have no ABO on your antigens on your blood cell surface. There's a lot of other antigens on there like M and N, which the book talks about and there's problems on, and also rhesus factor. So you're like A plus, or the plus or minus after your blood type is whether you have this rhesus factor or not. That's just another gene and another allele, and either you have it or you don't. But we're not going to go into that with this. How many of you guys have ever been, hopefully not, but anybody ever been to the emergency room and needed like a blood transfusion or anything like that? Did you know your blood type? You told them, didn't you? Yeah. You had it on your dog tags, so that's a guarantee. They did your blood type anyway, didn't they? Because if you kill somebody, then Alexander Shannara comes for you, right? So this is a thing where I'm making a joke about it, but if you're in the hospital and in the ER and you give the wrong blood type, even if the person is 100% sure, you are going to kill them, almost guaranteed. The reason is because when your immune system is developing, you have the capability to make antibodies that fight against invading bacteria, viruses, what have you. And the antibodies do a really good job at recognizing proteins. They can even to some extent recognize these, or to a good extent recognize some of these sugar molecules like we have here. But as you're developing, your immune system learns what antigens you have on your own cells, and you suppress those. Otherwise, your immune system would kill you. So autoimmune diseases is what happens when that goes awry a little bit, but not badly enough to kill you. It's your own immune system attacking yourself, right? So a person with blood type A would have A antigens, and so their body would not be making antibodies to A antigens. But because they have no B antigens on their red blood cells, if they ever encounter the B antigen, boom, antibodies get made. How would they encounter the B antigen? If they go to the emergency room and they either get type B blood or type AB blood, both of which have the B antigens. So if this type A person gets type A blood, there's no problem. But if they get whoops, hold on. This is the recipient to the reaction to that. Okay, so if the type A person gets this is the recipient. If the type A person gets type B blood, then they're in trouble, and you get a reaction, right? And you get it clots up the blood, and you're dead. So if you are type A and you get type B or AB, problem. If you're type B and you get type A or AB, you have a problem. The only person that can get any blood type is type AB. You notice they don't react to anybody. The reason is because they have the A and B antigens, both of them, and they don't make antibodies to anybody. Me, the type O over here, will react to everybody except another type O. So if you are blood type O, you can only get a transfusion from another blood type O. That would be bad, except that blood type O is the most common. It's like 36 or 37%, so we're good. And blood type O is also the universal donor. So anybody, well, I think I'm positive. So taking the rhesus factor out of the equation, With regard to AB, my blood is, if I donate it, is going to be able to be used by anybody. So we're the universal donor. Type AB is the least common. I think it's only like 6 or 7%, something very low like that. But they can get blood from anybody. So it works out, right? All right. Oh, I should say they're out there taking blood today. And you can get, oh, come on. You can get a nice t-shirt for donating. And they also give you nice drinks and cookies. I'm going out there afterwards. So if anybody wants to join me, I'll race you. We'll have them stick us at the same time. I'll be squeezing that little thing. Come on. It's no fun unless it's a contest, right? Okay, so whoops. So codominance is present in some cases when you would think that complete dominance is there, but it all depends on your perspective. So let's first talk about cystic fibrosis transmembrane conductance regulator, CFTR. So someone who has cystic fibrosis has a defect in this. Cystic fibrosis transmembrane conductance regulator is a chloride ion channel. If you have a defect, you don't regulate chloride ions very well, particularly in the epithelial cells of your respiratory tract. This is a problem because if you don't regulate chloride, you don't regulate sodium that well either, and you get mucus buildup. Just like it's not the HIV virus that kills people who have HIV and then get the disease AIDS, here, having this doesn't kill you. It's the buildup of mucus which allows for infections by microorganisms that normally would not be a problem because normal people can get rid of the mucus. So this is a recessive allele, and so you have to have 2 copies of it. But it is prevalent mostly in Caucasians of Central European descent, right? And there's a reason that it's I guess popular is a wrong reason. There's a reason why it's prevalent. Has anybody heard of this before? Nope. All right. So, what was killing off people? Name a disease that's not the plague that killed off a lot of people in Central Europe. If you get what? Nope. Not polio. I'll give you a hint. If you get this disease, you lose a lot of water through your digestive tract and you essentially diarrhea to death. Yes, there you go, cholera, right? So cholera was a big problem, infected water supply, right? And you would pick it up out of the water supply. So even though now people with cystic fibrosis now have a heck of a lot better prognosis because they have protease drugs that will bust up the mucus, and there's also interventions where they, like bend you over and whack you good, to break it up, literally to allow it to be coughed up and stuff. It's a lot better than it used to be, but at this time, if you got this, let's just say you weren't getting to reproductive age, right? However, when cholera was killing off 75% of the population at every village, all of a sudden we got a winner over here. So heterozygotes, even though they appear healthy, have a somewhat elevated survivability to cholera. So it's one of those things where there's 2 selections, one going for and one going against. If you're a heterozygote, you don't die like the rest of your village does. So that means that these people have a significant survival advantage. That survival advantage is well higher than the penalty that you pay where a fourth of the progeny of a heterozygote mating will get the disease and be dead. So even though it's not a problem anymore, I think it's 1 out of every 2,000 or 1 out of every 2,500 Caucasians, from that area have this allele. All right, so if you are a heterozygote for this, you would appear healthy. Forget the advantage because it's not, visible. But if you were to go down on the molecular level, you would see that half of the receptors are good and half of the receptors are defective. The reason why it looks like complete dominance instead of codominance is your perspective. You can't see down inside the epithelial cells and look there. Let's look at Dumpy and Unk here, right? So we're going to be working with those guys today in the lab. Some of you saw them last week and some of you didn't. The Dumpy worms are the ones that are short and fat, and the Unk worms are the ones that are, Unk means uncoordinated, so they are mostly paralyzed. They can move a little bit, but not very much at all. In these cases, so the Dumpy that we're working with is a problem with collagen. So collagen helps to hold the integument together. So essentially the skin of the worm, or the outer covering of the worm, and attach it to the muscles. And Unk are various things. So some of the Unks we have make defective muscle proteins, and some of them, I know Unk13 and chromosome 1 is like this, makes a defective neurotransmitter, so the motor neurons can't signal for the muscle to go. In all the cases of what we're going to do in lab, wild type is dominant over the mutant. So to be Dumpy or to be Unk phenotype, you need to be Dumpy over Dumpy or Unk over Unk respectively. But if you went and looked at the molecular level, then you would find out that you would make half normal collagen and half defective. But half normal collagen is enough to make the worms look normal. And if that's what you're looking at, is just looking at the phenotype of the worms, then it would appear dominant. I'm just trying to make sure you understand that dominant or recessive is dependent on your perspective. It's what we're looking at. All right. So now we're going to talk about penetrance and expressivity. So these can mess around with the phenotype ratio that you would expect. When you have penetrance and expressivity, what occurs is that not every individual with a certain genotype will get the phenotype that you would expect. And the reason for that is because you have to take into account the whole rest of their genome, all the other genes that they get. Right? So we're using here polydactyly as the example because that has both penetrance and expressivity issues. So polydactyly will result in like extra fingers or extra toes. This is common. Again, go back to the cat example. What? Yeah, but come on. Anyway, well, I don't have a picture of that. I should look. So sometimes the polydactyly is fairly common in cats, where they'll have like an extra claw, or extra toe and claw on their foot. And these are sometimes referred to as Hemingway cats. If you've ever been to or ever go to Key West in Florida, there are a heck of a lot of these running around because Ernest Hemingway, the famous author, lived in Key West and liked this kind of cat. So there's a lot of feral cats running around the street, just wild, and people feed them that have this phenotype, and it's dominant, so it's easy to keep passing on. You do see them in if you look in like the Humane Society pictures of cats available for adoption and whatnot, you see people with them, and oftentimes somebody will get it and they'll name their cat Digit because it's got an X, right? I've seen a lot of Digits. Okay, so penetrance. That's the percentage of individuals with a particular genotype that express the phenotype at all. you can add to that. So if you have 70, I don't know what this is, but let's say the penetrance for polydactyly is 75%. That means only, and it's a dominant allele, that means only 75% of the individuals who have a dominant allele for polydactyly will actually show it. The other ones will not at all. They'll look perfectly normal. But they'll essentially be an invisible carrier of that. Not because they're heterozygous and it's recessive, but because the phenotype didn't show for whatever reason, for their other genes. Expressivity, the degree to which a character is expressed. So if you have only variable penetrance, let's go back to that one, either you got it or you don't, and there's a certain percentage. If you have variable expressivity only, everybody with the right genotype has that phenotype, but there's a difference in level of expression. And that happens here too. So in this case, you see, like, look, man, there's an extra toe there, and it looks legit, right? So, but there's all kinds of differences where you can have from a full extra toe to everything down to like a little stumpy thing that's barely there. That's difference in expressivity. But polydactyly shows differences in penetrance and expressivity. So some people who have the polydactyly allele don't get it at all. Of those that do get it, there is a variation in the severity of it. All right, so I've made this example that I hope will help you. So every one of these diamonds has the allele, the dominant allele, that makes you a blue diamond. Right? If you have variable penetrance only, then even though every one of these diamonds has the blue diamond allele, some of them are blue and some of them are not. But every one of them can pass the blue diamond allele on to their progeny, just like if you had the phenotype. If you have variable expressivity only, you will notice that there are some diamonds that are as dark as the darkest ones, but there are some that are fairly light as well. However, because there's only expressivity, every diamond is blue. It's just you're blue to a different degree. If you have variable penetrance and expressivity, some of the diamonds are blue and some are not, like completely white. And of the ones that are blue, you get a variation in the level. Make sense? All right. Now we're going to go back to the lethal alleles. This is an old story because these yellow mice were part of the Victorian era mouse fanciers that I was telling you about earlier, and people always wanted to get what they refer to as sports. So if you're breeding plants or animals or whatever, a sport is something that comes out of a cross that's got a completely new phenotype, and then you try to get it and continue to breed it because you've got until everybody gets it, you've got something unique. They found yellow mice, right? They determined from crosses that yellow was dominant to other colors like gray or white, whatever. But no matter how many crosses they set up with yellow mice, they could never get a true breeding line. They always got progeny in there that were not yellow. And the reason why this occurs, they eventually discovered when scientists ran the numbers, is because even though the big Y allele here is codes for yellow, so yellow is dominant over non-yellow, having two big Ys is lethal, meaning that you die embryonically. You never go beyond this stage of development, and they may not even be born dead. They may be reabsorbed by the time the rest of the litter is born. So that's going to skew the ratio of colors here. Just like when you took out the people with the disease, it messed around with it. So here there are 4 squares in your Punnett square. It's a 1 to 2 to 1, so 2/4. So 1 to 2 to 1. But progeny that died during the pregnancy and are never born are not counted. Whoops. And so what would happen is, let me go over here, that you're going to change the phenotype ratio. All right, so normally you would expect a 1 to 2 to 1 phenotype ratio. Here's 1, there's 2, and here's 1, right? But what happens is this guy is dead, so it doesn't get counted. You still get 2 of the heterozygote, which is yellow, and 1 of the little y homozygote, which is, say, gray or white, just something other than yellow. But now instead of a 1 to 2 to 1 phenotype ratio, it's just 2 to 1. So it's 2 of 3 and 1/3 gray, right? So because the yellow mouse is taken out, it never gets counted. And we're talking about the progeny. I mean, if you look at it, when I go back to the slide, if you look at the slide, they just never get counted. I had a student one time that argued, well, if you open it up, after the exam, of course, if you open it up and looked in utero, you might be able to see one with yellow, so you could have counted. I'm like, no. Look at the picture. It doesn't have fur yet. It's, you know, I said it's dead to start with. So it's not counted. So 1/4 of the progeny never get counted. So the ratio is changed. If this did not have the lethality associated with big Y, the yellow color, then you would expect a 3 to 1 phenotype ratio. But here you only get a 2 to 1 phenotype ratio. All right, so I'm always interested in finding out how things work. And so even though this stuff was done fairly early on, like in the early 1900s, it wasn't until the 1990s that somebody actually cloned or discovered what the yellow allele looked like and went in and saw what was going on. So we don't get to molecular biology until the second half of the course, so I'm going to have to give you a crash course explanation here. So here's a gene for yellow fur color. There's a little sequence of DNA ahead of the gene called the promoter that acts like a switch. This DNA gets transcribed into RNA and translated into protein. This DNA does not, but this DNA contains binding sites for proteins that are required to get it turned on. So in the wild type mouse, the little y allele, the yellow switch is fine and the yellow gene is over there. And then there's another gene right next door to it called MERK. MERK is an essential gene. That means that if you have 2 defective copies of this, you die. You don't develop. But wild type is dominant, so all you need is 1 good copy. In the yellow mouse allele, here's what happens. There's a deletion. So a deletion is when part of a chromosome, for whatever reason, has gone missing. Just like when we talked about in lab, you get the inversion where a piece comes out, flips, and goes back in, or the translocation where pieces of chromosome break and switch between non-homologous chromosomes. Here, what happens is the chromosome breaks in 2 spots, And so the chromosome, the good chromosome, breaks here and there, and in the emergency situation, the cell's like, I got to fix this fast or I'm going to die, and it goes to a last resort mechanism where it adds this part down to this part. Where it says deletion, that's not there anymore. So here's what happens. The yellow is only supposed to be expressed at certain times during the hair's development, like when it comes out of a hair follicle. So you're only supposed to get like a little line or ticking on the hair. It's supposed to be the normal color and just like a little, a little yellow splotch on it. But what happens is this part of the switch is what turns it on and off at the right time. This part is just has it on. So what happens here is the switch is broken, it's stuck in the on position, so that the whole hair gets yellow colored instead of just a stripe. Also, a big part of the Merck gene is gone, so if you're homozygous for that, you won't develop. So what I'm showing here on the slide is the heterozygote, that's the yellow mouse. Because this is broken, this is a dominant allele. It doesn't matter that that's regulated normally. This is on all the time, so the entire hair of all the fur is yellow. But because in the heterozygote you have one good copy of the Merck gene, they get to live. If you are homozygous for this chromosome, you have 2 copies of this one up here, you are the non-yellow mouse because regulation of the yellow gene is fine. If you get 2 copies of this, that's the big Y allele down here, then you're in trouble. You should be still yellow fur, but because you're missing a big piece of the Merck gene, you're dead. So this allele, a deletion allele, actually affects 2 different genes, and that's why you get 2 effects. You get the dominant yellow because it takes out this part of the promoter or switch, and you get recessive lethality because it takes out a piece of this Merck gene that's required for development. Everybody's quiet. That makes sense? Good, good, good? All right. All right, so when you have lethal alleles, go back here. When you have lethal alleles, somebody that should be there as part of the phenotype ratio is missing. You can tell when you have a lethal allele because the ratio is skewed. Let's look at this now. In fruit flies, for wing shape, big C, curly, is dominant to wild type. So curly wings or straight wings is little c. And also hairy body, big H, is dominant to hairless. These genes are on different chromosomes, so if you do a heterozygous cross, it's just like the dihybrid cross we talked about back in chapter 3 with the round and wrinkled and yellow and green peas. So you should get what kind of a ratio with the phenotype when you do a dihybrid cross? 9 to 3 to 3 to 1. You got it. All right. So we get a 6 to 2 to 3 to 1 phenotype ratio. Now here we're talking about a lethal allele. Coming up, we're going to talk about epistasis. In epistasis, the 9 to 3 to 3 to 1 phenotype ratio is going to get changed, but nobody is going to be missing. You're just going to combine different classes, and we'll see what that looks like. But here, with lethals, something is missing. So instead of a 9 to 3 to 3 to 1, you get a 6 to 2 to 3 to 1. So the first 2 here have missing alleles, right? The answer is, the question I'm asking is, which of these alleles is going to be lethal? So it's going to be either C or H in either big C homozygote, little c homozygote, big H homozygote, or little h homozygote. What we want to do is eliminate the possibilities, right? So which of these four classes has the correct number of members for the ratio? Which of these? Yep, this one and this one, right? So obviously, because all of these are here, we can eliminate what they are. So what's the genotype of the wild hairless going to be? Little c, because it has to be homozygote. Yeah, little c homozygote, little h homozygote. So if either little c or little h homozygote was lethal, then this wouldn't exist, would it? So we can eliminate little c homozygote and little h homozygote as possibilities, right? Yep. You with me? All right, so 6, curly, hairy. So curly is dominant and hairy is dominant, right? So what is the genotype of this class going to be? So, well, big C, big H, but there could be also heterozygotes, right? So this is going to be big C blank, big H blank, right? Then the two curly hairless, what's the genotype of this one going to be? That's a capital H. Then 3, that's going to be, what's this one going to be? Whoops, sorry. I said the right thing and you guys said the right thing, and my brain, I'm talking and writing at the same time is not something I excel at. So this one is going to be the opposite, right? Little c, big H blank, right? And then finally, wild and hairless, those crazy flies. Right? So because these two have the right number of members in the class, we can rule out little c and little h as being lethal, right? So let's move up to the next one from the bottom. So what else can we rule out from? What can we rule out from this one? What? Homozygous for which H? Big H, right? Because the bottom, we've already determined that homozygous for little h is fine here, but this one can be big H, or big H, little h, but because you see them all, big H must be fine too. So what's left? Right. So that's so far that's what it looks like, right? It's going to be big C based on what we eliminated. However, we're going to also want to check ourselves to make sure we're right. So do both of these up here have big C as part of them? Yes. So that makes sense, right? So everybody who's So there's 3. If you write out everything that goes with this one, all the possibilities, 3 of them will have big C, big C. And that's why 3 out of the 9 are gone. So there's only 6 left. So the only ones of these that live are the ones that are big C, little c heterozygote. Same with this one. This should have 3. There's going to be 1 big C and 2 big C, little c heterozygotes. Those live. The big C dies, so you go down from 3 to 2. Explain how I knew that. Oh, so we started from the bottom. So because this one is alive, we know that little c isn't lethal, because otherwise there wouldn't be any. We know that little h isn't lethal, right? So then we go up to the next one. This guy has all 3 of them that's there. We've already shown that little c isn't it. And this one can have either big H, or big H, little h. But we've already shown down at the bottom here that little h isn't it. So now we're like, okay, the third one of these is big H, and because you see all the members of the class, then big H can't be it either. Now in these 2 at the top, we've already shown little h can't be it. We've already shown that anything with the h's can't be it, right? So, and we've already shown down here that little c can't be it either. So the only thing that's left from these is big C. And sure enough, they're missing things for that class. Yep. Okay. We good? Okay, so we're talking about, let me recap a little bit. So we talked about lethal alleles here. When there's a lethal allele, there are members of classes missing. When we did incomplete dominance, there were no members of the classes missing. You went from 3 to 1, which is 4 total, to 1 to 2 to 1, still 4 total. Here, we're going to talk about epistasis. In epistasis, you're going to get a 9 to 3 to 3 to 1 phenotype ratio. How many squares are in a Punnett square for a dihybrid cross? 16. And 9 to 3 to 3, 9 plus 3 plus 3 plus 1 is 16. So the way you tell whether there's a lethal allele, like we just talked about here, or whether there's epistasis, is whether there are some members missing from the 9 to 3 to 3 to 1, or in the case of epistasis, you're just going to get, there's still going to be 16 total, it's just the phenotype classes are going to be mixed up. Make sense? So you were asking for the cats, here's the, it's America's dog, right? The Labrador Retriever, everybody's favorite animal, just about. All right, so we're going to talk about lab coat color. What are the coat colors in Labrador Retrievers? There's black lab. What's this guy? Yellow lab. And what's the other one that's not shown here? The brown, the chocolate lab, right? So there's 3 colors. The black, brown, and yellow Labrador Retriever coat colors are controlled by 2 genes, the B gene and the E gene. If you take a dog that is homozygous, dominant for both, black color, one that's homozygous recessive for both, yellow color, cross them, 100% of the progeny will be heterozygous. Big B, big E, little e, right? And they will all be black coat color. However, if you take these guys in the F1 generation and find somebody else's dog somewhere else that had the same cross, so you're not crossing litter mates, which is not a good idea, Then in the F2 generation, this guy shows up. So you get 9 black, 3 chocolate, and 4 yellow. So the 9 to 3 to 3 to 1 ratio has been collapsed. There's a class missing. In all these epistasis cases, the reason why there's a class missing is because 2 of the classes have been combined. So you would expect a 9 to 3 to 3 to 1, right? You get a 9 and a 3, but instead of a 3 and a 1, you get this guy here. So you can see that this is epistasis and not a lethal allele because you add them up and they're still 16. 3 plus 1 is 4. So what's going on? In the cases of epistasis, unlike the case in the P color and P shape, where the color doesn't inhibit your ability to tell the shape, and the shape doesn't inhibit your ability to tell the color, here both these genes are involved in coloring the fur. And so what happens with one is going to affect the other one in the phenotype. Here's what really happens. So 9 are going to be the, as you would expect, big E blank, or a big B blank, big E blank. 3 are going to be little b, big E. And let's stop right there. The B's are for the black and brown coat color, or black and chocolate. Big B is dominant to little b. Big B is for black, little b is for brown. But you notice something about everybody from where I'm drawing this line up is that they both have a big E blank. These 2 down here both have little e. And so what you can see is here up at the top, when you have at least one big E, big B blank means black, little b means brown, but here where you have little e, doesn't matter what you get for the B's. You're going to be a yellow lab no matter what. The reason for that is epistasis, and we'll go back to this in just a minute. All right, so epistasis is a gene interaction event. Both genes are involved in specifying the same character, in this case coat color. When epistasis is at play, an allele at one locus, or one gene, masks the effects of an allele at a second locus. So here comes your terminology. The epistatic gene is the gene that does the masking, and the hypostatic gene is the gene that is masked. It is very crucial at this point that you do not confuse epistatic and hypostatic with dominant and recessive, because they are not the same thing. In the lab example, the little e allele is epistatic to alleles B and B, because if you have little e homozygote, it doesn't matter. You could be big B, big B, big B, little b, or little b. You're going to be the yellow cream colored lab no matter what. The hypostatic gene is the B allele. So like I said, black coat is dominant to brown coat, but big E allele is required for the ability to deposit any pigment in the hairs, either black or brown. So, yes? You could say that, but I really, really don't want you to use the word dominance here. So one locus, the one locus masks the effect at another locus. The masking one is the epistatic one. It's the one that is better than the other. I guess it has more power than the other one. I just don't want you to use the word dominant because that's not what it means. It's going to get confusing. And if you put dominant on a thing, if you say dominant when you really mean epistatic, I have to take off for that. All right. So This is the first example of epistasis. It's called recessive epistasis. So epistasis is where one gene masks the effects of another gene. Recessive epistasis is where, in order to show the epistasis effect, you have to be homozygous recessive. So you have to be little e. You have to be little e to show the effect. So if you are little e, this is epistatic over big B blank, also epistatic over little b. It doesn't matter if you would encode black or brown fur color. If you can't put the color into the hairs, it doesn't matter what that color was going to be. It isn't going to happen. Make sense? Let me do one more and then I'll come back and hit this again on Thursday. I want you to please go ahead and try to read through the rest of this before Thursday so that you can get a head start on understanding. All right, so dominant epistasis. Same thing, but the epistatic gene is dominant, so you only need one allele. So in this case, they're using squash coloring. They have a true breeding white and a true breeding green squash. Cross them into the F1, you get all white, so it looks like white's dominant. But then, just like in the F2 generation, the chocolate lab appeared where it wasn't in any of the parents of the F1. Here, now in the F2 generation, you get a 12:3:1 phenotype ratio. How can you tell that this is epistasis and not a lethal allele? Yes, both of you are right. They add up to 16. There's nothing missing, they're just regrouped. And what's regrouped in this case? The 12 is regrouped. It's a 9 and a 3. All right? So here's what it is. I'll stop here because I want you to go, but look. So in this case, there's W and Y. W, if you are big W, then you are going to be white, no matter what you have for the Y's. But if you are little w, then you get a 3:1 phenotype ratio there. So big Y for yellow is dominant over little y for green, but in order to see the yellow or the green color, you have to be little w. More on this, and I will go back and do it again on Thursday. So see you at lab today. Maybe somebody will decide they want to drain themselves out here. You can go out and it really, if you've never done it before, it really doesn't hurt. You can come out and watch me get drained and laugh. Oh, so but just you can't take they for whatever their reasons, if I'm out there, you can't take pictures. They don't like that. So all right, see you later. Make sure to read through the rest of this.